Example 1. If the bark and phloem of a woody shoot are peeled off by ringing, the whole plant will eventually die because A. Water does not reach the leaves. B. Water and salt remain below the ringed portion. C. There is withdrawal of water from the root by soil. And D. Manufactured food does not reach the roots. To answer this question, you must note the conducting tissue that is removed, and that is phloem. And the work of the phloem is to conduct manufactured food from the site of production, that is the leaves, downwards to the roots. So if the phloem had been peeled off, manufactured food could not reach the roots. So answer to the question is D. Number 2. Which of the following is not a characteristics of monocot plants? A. Occurrence of secondary thickening. B. Parallel venation. C. Scattered vascular bundles in the stem. D. Floral parts arranged in trays. Now, the floral parts of a monocot plant is always in tray or multiple of tray. When you cut the stem, a transverse section of the stem of a monocot plant, and you examine the vascular bundles, the internal structure, the vascular bundles are scattered. The leaves of monocot plants like grass, maize and so on, you see the veins are arranged in parallel to one another. So that's correct. It's A that is not correct. They say occurrence of secondary thickening. And secondary thickening is made possible through meristematic tissue of carbion. And there is no carbion in monocot plants. So answer to the question is A. Number 3. In lower plants like mosses, the structure which performs the function of the roots of higher plants is root hair. That's A. B. Hypha. C. Rootlets. And D. Rhizoids. Root hair is not found in lower plants because you have to get 2 roots before you can get root hairs. Hyphae is just a vegetative part of fungi. The rootlets are the secondary roots or tertiary roots. So when the actual answer here is rhizoids, because they specifically mention moss here as well. So answer is rhizoid. 4. The correct sequence of tissues in the anatomy of young dicotyledonous stem from the inside to the outside is A. Pith, phloem, carbium, xylem, cortex, and epidermis. B. Xylem, phloem, carbium, cortex, endoderm, cholechyma, and epidermis. C. Phloem, xylem, carbium, cortex, endodermis, cholechyma, and epidermis. D. Pith, xylem, carbium, phloem, cholechyma, parenchyma, and epidermis. This type of question you have to understand the anatomy of the stem. And so let's look at the diagram. This is the transverse section of dicot stem. Now, from outside we have epidermis. The epidermis, after epidermis you have cortex. This area is cortex. Now, the cortex, if you look at the tissue side the cortex, you can find cholechyma, then a wide middle area that is parenchyma. Then towards the inner core, you have sclerenchyma that may make up the pericycle, but in some plants it's separated from pericycle. So pericycle, and in some plants the pericycle is endodermis for storage of food. Then when you enter the inner core, you have the vascular bundle. The vascular bundle is made up of the phloem, then a ring of meristematic tissue, the cambium, and the xylem. Then inside we have the pit. Now the question is the arrangement of the tissue from inside outward. So you have to be pit. After pit, you enter the xylem, xylem to cambium, cambium to phloem, then the pericycle or cholechyma, then the parenchyma, then you now enter the epidermis. So let's see the one that fits in this description. Option A, pit, phloem, jump the xylem. So option A is gone. B, it starts with xylem instead of pit. So B is gone. C, start with phloem. That is, it jumps the pit and xylem. C, it doesn't work. Then you come to D, which is obvious answer. Pit, xylem, cambium, phloem, cholechyma, parenchyma, and epidermis. So option D answers the question. Number 5. The sclerenchyma cells are lignified to A, strengthen and support the plant, B, transport synthesized food, C, conduct water and salt, and D, protect the plant from injury. Sclerenchyma cells are lignified, meaning they are thickened through the deposit of calciums and other materials on the inner wall, and to the extent that it crushes the protoplasm and the cells are dead. In that case, sclerenchymatous cells are only useful for strengthening and support of the plant. So A is the answer.